name is Ed Boyden. I direct a group at MIT that works on new tools for observing and controlling complex biological systems. Today, I'm very excited to tell you about a technology we developed that lets you do nanoimaging, nanoscale resolution imaging, on conventional microscopes. This is important because biology is fundamentally about nanoscale building blocks, gene products, biomolecules, and so forth, that are often organized with nanoscale precision throughout cells and tissues. But how can you image with nanoscale precision? That's a difficult thing to do. Well, today I'll tell you about expansion microscopy, a method that lets you do just that. In expansion microscopy, we do something very different than the past 300 years of imaging where a lens magnifies an image of an object. Instead, we take an object and physically magnify it itself. We do that by chemically weaving a dense spiderweb-like mesh of swallowable polymer, basically the stuff in baby diapers, inside cells and outside cells, in between biomolecules and around biomolecules. We anchor key biomolecules to the polymer, chemically soften the specimen, and add water. The swallowable polymer absorbs the water and swells, but because of the little handles or anchors, all the biomolecules are pulled apart. This idea owes a debt to several old lines of research. In 1980, for example, Professor Toyoshi Tanaka at MIT was working out the physics of swallowable polymers, like sodium polyacrylate. Water is drawn in through osmotic force, and the polymer threads are pushed apart from each other. The polymer threads are highly charged. Sodium polyacrylate is chock full of carboxyl groups, and so the polymer swells enormously, 1,000 times or more in volume. Now, you can't just dump the polymer on top of the specimen. How do you weave that dense spiderweb-like mesh of polymer inside the cells and outside the cells, in between the biomolecules and around the biomolecules? Well, interestingly, around the same time in 1981, Christine Dreyer and Peter Hausen published a paper where they took preserved biological specimens and chemically synthesized a hydrogel throughout them. They used an uncharged hydrogel, polyacrylamide. We're using a highly charged hydrogel, polyacrylate. But it's interesting that if you think about it, if you put two and two together, maybe what I'll tell you about today could have been developed nearly a half-century ago. In the case of Hausen and Dreyer, they took these preserved embryos and tadpoles, formed a polyacrylamide hydrogel throughout them, and then showed that they could do immunostaining and imaging of these preserved specimens. So the basic idea then is we want to take a cell like the one on the left and turn it into something like the cell on the right, a constellation of biomolecules hovering in space, their relative organization preserved. Two biomolecules that used to be touching are now some minimum distance apart, and two biomolecules that are some distance apart will be scaled up by some linear factor. Now, to make this work, we had to develop several chemistries. In this cartoon, one of them is illustrated. Biomolecules, in this case proteins, are shown in brown, and we developed handles or anchors that bind to the biomolecules, here shown in purple. In addition to handles for proteins, we also developed handles that bind to DNA and RNA and other kinds of biomolecule. Next, we have to weave that dense spiderweb-like mesh of swallowable polymer. We had monomers, shown as little white spheres here, and they self-assemble into polymer chains through a process called free radical polymerization, not unlike what Hausen and Dreyer published in 1981. When these growing polymer chains encounter a handle, they form a covalent bond. Finally, we soften the specimen using heat, detergents, or enzymes, and then when we add water, the polymer threads swell apart from each other as they absorb the water, but this time, because of the handles or anchors, the biomolecules are pulled apart from each other. By design, this expansion process is as even as possible. Panel B shows a piece of the mouse brain before we expand it. Panel C, the same piece of mouse brain after we expand it. It's 100 times bigger in volume. about 4.5 times bigger in each direction. The polymer starts out very compact. The spacing between polymer threads is just a couple nanometers, and at the end of the day, it's expanded into an extended form. Here's a little time-lapse video of the expansion process. We polymerized this piece of mouse brain earlier, and right there we add water. It's sped up for an hour to a minute, but I hope you can see that this piece of the mouse brain is expanding before your very eyes. So by design, the polymer is very dense, the specimen is softened. We designed the expansion process to be as even as possible. But this is biology, we have to prove it. And so in the early days of expansion microscopy, we spent a lot of time taking pre-expansion images with a classical nanoscale imaging method, like a super resolution microscope. And then we would take post-expansion images with a conventional confocal microscope. The distortion is not zero, but it's really small, just a few percent over a typical microscope field of view. Turns out, for the vast majority of biological and medical questions we are facing right now, that's what we want, to see the relative organization of biomolecules. In addition to measuring the distortion, we can measure the resolution. We can take specimens like these hex cells, where the microtubules have been decorated with antibodies against tubulin. The antibodies are bearing fluorescent molecules. We can take our images and deconvolve them by the ground truth, which in the case of microtubules, we've obtained as a community over many decades of classical nanoimaging, like through electron microscopy. And we can measure the resolution. So we get a resolution of around 65 nanometers. Now, if you think about it, a typical 300 nanometer lens, if you expand 4.5-fold, you might expect to have a resolution of 300 divided by 4.5, or about 65 nanometers. That raises the question, if you expand more, would the resolution become even better? As I'll show you later, that's true. But even with 4.5-fold expansion, there are many things that we can do that are difficult, if not impossible to do with earlier means. Here on the left is a piece of the mouse brain, cortex, hippocampus. Each white square is zoomed into in the image below. The color code is shown on the left. These are fluorescent antibody stains. As you can see, the right-hand side, these are the same fields of view as the left-hand side, but after we expanded. And there's a lot more detail visible. In fact, we can see synaptic connections. The blue is a presynaptic protein, and the magenta is a postsynaptic protein. And we can measure the distance between these pre- and postsynaptic protein densities. In fact, we get exactly the same answer, shown in the upper right, as Catherine Dulac and Jiawei Zhuang measured many years ago with storm microscopy. But this time, we can take these images on a regular confocal microscope. So expansion microscopy really democratizes nanoimaging. Anybody can do nanoimaging with existing microscopes that are commonly. By the way, this first paper I forgot to mention was spearheaded by Fei Chen and Paul Tilburg when they were graduate students in my group. Here in the upper right, you can see antibody labeling against mitochondrial and lysosomal proteins. At the bottom, labeling myelin basic protein with an antibody. This is work done by Ray Gao, Sho Asano, and Gokul Padula, working between our group and Eric Betzig's group. You can express artificial proteins in these brain cells. So for example, here we are expressing fluorescent proteins derived from jellyfish and coral randomly in brain cells using viruses. If we zoom in, of course, quickly we hit the resolution limit of the microscope and we cannot see the fine wiring of the brain. But after we expand, we can cleanly resolve the axons and dendrites of brain cells. So many groups are using some kind of color code plus expansion in order to map brain circuitry. So far, everything I showed you is with protein labeling, but you can also label other molecules like messenger RNAs or other RNAs. Here's work done when Oswasi joined our group, and a bunch of pre- and post-expansion images are shown here, where after we expanded, we did in situ hybridization with fluorescent nucleic acid probes. Here at the bottom are long non-coding RNAs like EXIST and NEAT1 being visualized. We can also visualize individual mRNAs in intact brain circuitry. Here we've anchored and expanded both proteins. You can see yellow fluorescent proteins shown in yellow, and RNAs. You can see messenger RNAs shown in magenta, labeled through post-expansion in situ hybridization. We can also do multiplex imaging. We can bring in libraries of hybridization probes, dozens or even hundreds at a time. Each hybridization probe has a little barcode on the side of it that we can then sequence. When you sequence something, you're just copying the nucleic acids. As you copy the nucleic acids, you're blinking out the genetic code, because each nucleic acid that you add, each nucleotide that you add, is bearing a fluorophore. Here you can see we took a couple dozen genes from the Allen Institute single-cell RNA-seq dataset. We did hybridization against several dozen of these in intact brain tissue and sequenced barcodes on those hybridization probes. The clusters we found are in the upper left. As you can see in the lower left, they agreed quite well with the classical cell type clusters. Our clusters are on the y-axis, the old ones are on the x-axis. But most importantly, we can visualize these cell types superimposed over the actual brain tissue. Here's the mouse visual cortex, layer 1 on the right, layer 6 on the left. We can see those cell types distributed throughout the layers of the visual cortex. Now, because expansion gives you nanoresolution on a conventional microscope, we can map the locations of messenger RNAs in nanoscale compartments like axons, dendrites, and synapses. You can see this in the lower left, where different RNA gene names are shown in green, and you can see the morphology of the cell shown in gray. This is work spearheaded by Shahar Alon, Dan Goodwin, Anu Sinha, Azwasi, and Fei Chen. As a side note, we can do a similar in situ sequencing approach for DNA, not just RNA. In work we did with Jason Van Rosto's group and Fei Chen's group at Harvard, Andrew Payne, Zach Chang, and Paul Reginato showed we could insert little barcodes into the genome and then sequence the genome right there inside a cell. This is not yet used expansion microscopy, but hopefully we'll integrate it in soon. Okay, back to expansion. When J.B. Chang was a postdoc in the group, we polymerized and expanded a piece of brain tissue, then formed a second polymer in the space opened up by the first expansion and expanded it again. If you expand 4.5-fold twice in a row, you get a total expansion of 4.5 times 4.5, or about 20-fold. Recently, Deblina Sarkar, Jinyan Kang, and Azwasi, with great help from Margaret Schroeder, and Amuchi Emanari and Christina Kitko made important versions of this, we showed we could do this double expansion technique with all off-the-shelf chemicals. Importantly, this offers a second advantage, shown in the cartoon. In the top row of the cartoon, if we bring in fluorescent antibodies to label proteins, they can label outside proteins around a protein complex, but they cannot get in. The green and the red antibodies can bind to the outside of a protein complex. But if you pull the proteins apart, as shown in the lower part of the cartoon, now the yellow and blue antibodies can access proteins inside the complex. We've decrowded the proteins and made more room for their staining. We decided to investigate synapses and other densely packed proteins in intact cells and tissues. Here you can see some synaptic proteins, shown in B, C, and D, are very densely packed. Panel B is a calcium channel, and panels C and D are pre- and postsynaptic scaffolding proteins. Yellow is pre-expansion staining, and there's not much yellow, but magenta is post-expansion staining. We get a lot more detail. Not all proteins are so crowded. F, G, and H show proteins that are very well stained before expansion, shown in yellow, and after expansion, shown in magenta. Since we saw a calcium channel being unveiled by decrowding, we wondered, can we understand how these calcium channels are organized at synapses? Collaborating with Tom Blampied's group, we showed that these calcium channels are not randomly distributed, but they're precisely coordinated with respect to other pre- and postsynaptic proteins. To make a long story short, these proteins are all organized together, perhaps to mediate the precision of synaptic transmission. Working with Li Wei Sai's group, we wondered, could we look at pathological proteins? Looking at the 5x FAD Alzheimer's model mouse, we did pre-expansion, shown in yellow, and post-expansion, shown in magenta, staining for A beta 42. with antibodies against A-beta-42, a major component of amyloid plaques. As you can see, post-expansion we saw much more detail than pre-expansion. In fact, we even saw some new things we didn't see before. These purple dots, amyloid dots that form almost periodic patterns. Those were only visible after expansion. This held for multiple independent antibodies against A-beta-42. And we never saw these periodic amyloid dots in wild-type mice. So far, every polymer strategy I've shown you is using free radical polymerization. But free radical polymerization has some flaws. While it's a very good method, it also has some very fine-scale defects. Dangling ends, as shown in the upper left, loops, and inhomogeneities. So Ray, Gao, and J.U. in our group, we wondered, could we make a more regular polymer? Well, for many years, people have studied these diamond lattice-like polymers that form almost crystalline structures, as shown in the cartoon on the right. We wondered, these polymers are nearly defect-free. Maybe they could expand more evenly than the free radical synthesized polymers? We decided to use viruses as sort of nature's scale bar. We used this so-called tetragel, as we call it, to expand herpes simplex viruses made by Rachel Neve's group. As you can see in the upper left, the tetragel, and the left-hand side of panel C, resulted in very round expanded viruses. In contrast, the free radical synthesized hydrogel resulted in recognizable viruses, but with much more irregular boundaries. The population data is shown in G. The free radical synthesized polymer hydrogel resulted in expansions with about 15 nanometers of accuracy, but the tetrahedral hydrogel was able to give 9 nanometers of accuracy. Here's some data showing how the shapes of these viruses were preserved, and you can see here different measures, the ellipticity, the sphericity, and so forth. And to make a long story short, the tetragel expanded viruses were much more even in appearance and more accurately expanded in their shape than the free radical synthesized hydrogels. Now, many species tissues can be expanded. In work done by Yongxin Zhao and Octavia Ricoeur, we expanded human biopsies. On the left, normal. On the right, cancer-containing. The color code is shown in the upper right. And these are tissues from prostate, lung, breast, pancreas, and we did many others. And we showed we could expand formalin-fixed, paraffin-embedded, fresh frozen, and many other kinds of human biopsy tissues. So nanoimaging can now really be brought into the human tissue realm as well. Let's summarize. Over 250 experimental papers and preprints have already come out doing expansion microscopy. It's being applied to the human kidney, to the microbiome, to plant seeds, to motor proteins, to looking at how viruses spread from one cell to the next, to chromatin, mitosis, and the list goes on and on and on. You can find on our website, expansionmicroscopy.org, step-by-step cookbook-style protocols of how to do expansion microscopy. During normal times, we often would host people at workshops held at MIT or all over the world to learn how to do expansion microscopy. But now, of course, we cannot do that. And so last summer, we published a step-by-step cookbook-style tutorial of how to do expansion microscopy with great detail, even showing how to handle a cover slip with a pair of forceps, how to incubate the specimen to form the gel, how to pick up a hydrogel with a paintbrush, and more. So to summarize, we've discovered that we can physically magnify biological specimens. This is very different from the past 300 years of biological imaging, where a lens is used to magnify an image of a biological specimen. This technique, expansion microscopy, can be applied to all sorts of tissue types and to visualize all sorts of biomolecules, proteins, nucleic acids. The list goes on and on. This technology democratizes nanoimaging, and anybody can do nanoimaging by using it. Our website contains protocols so that anybody can practice it, and we're always happy to answer questions. I think along the way, I've acknowledged all the people in the group who have led these projects, but I just want to end on this slide because it contains an even longer list of people at the top who have helped with these projects, and an even longer list than that in the middle of the slide of collaborators all over the world who we've worked with to make these projects possible. Please visit our website, synthneuro.org, to read about protocols or how to access reagents, and do not hesitate to email me at edboyden@mit.edu if I can be of help in any way. We really want to help people solve their problems in biology and medicine, and given that biology and medicine are fundamentally, one might argue, about nanoscale building blocks and how they're organized, we would love to help you all solve problems using nanoimaging. And with that, I'd be happy to take questions.